Hypertonic saline inhibits SARS-CoV-2 replication by an intracellular effect and does not affect its interaction with human AC2. So what I'm gonna talk today? I'm gonna talk about the effect of the NSL concentration in the replication of SARS-CoV-2 using various cells. So it's an in vitro assay. Unfortunately, we don't have in vivo assay. Some hypotheses of how NSL avoid the replication of SARS-CoV-2 inside of cells. Possible general effect by avoiding respiratory virus replication. So maybe this mechanism, it's not only for SARS-CoV-2, but may be used for all respiratory virus. And an explanation of how SARS-CoV-2 spike protein keeps its interaction with human AC2 in low and high concentration of NSL. So we're going to talk these 3 topics today. Okay, so what do we know about coronavirus? So coronavirus, it is infecting different mammalian and also avian species. It's classified in genus known as alpha, beta, gamma, and delta. And in the case of SARS-CoV-2, that severe acute respiratory syndrome coronavirus 2 belongs to Coronaviridae family. It's the circles, as you can see in this electron microscopy picture, that was obtained in our group by the SARS-CoV-2 virus isolated from the second patient in Brazil. Are you listening to me? Okay. Okay. And this virus has a positive sensitive strained RNA and belongs to the genus of beta coronavirus subgroup 2. So this crown all around the virus, it is made of a protein called the spike protein, and that gives the name for the virus as a coronavirus because it looks like a crown. There are 7 strains of coronavirus that infect humans. 4 of them causes mild and self-limiting upper respiratory tract infection, or a symptoms very similar with common cold. And 4 of them are also found here in Brazil, okay? But 3 of them cause severe acute respiratory syndrome and outbreaks, as happened with SARS-CoV-1, that we call one, but doesn't have the one, that killed around 800 people in 2002-2003 in China. The MERS, Middle East respiratory syndrome, that killed 600 people in Middle East in the period of 2012 to 2014. And the actual pandemic situation caused by the SARS-CoV-2 that causes the COVID-19 disease that started in China in 2019 that everybody know that, and it killed more than 1 million people worldwide until now. So here it's like a cartoon representing the virus, the protein located in the virus. Here in green is the membrane protein, membrane of the virus, because it's a membrane, this virus has a membrane. And some structural proteins shown here, like the spike protein, enveloped protein, and some intracellular proteins like the nucleocapsid that interact with the RNA of the virus. And here shown different proteins that are it is from the virus, okay? Looking mainly to this protein, that is what we're gonna talk today, it's the spike protein. That is a surface glycosylated spike protein of SARS-CoV-2 that binds this. It's a human receptor known as an angiotensin converted enzyme 2 that is the human receptor. So it is like the main entrance of the virus in the human cells. It's interesting that these proteins are homotrimer, so there are 3 units of the same protein that have built like this spike all around of the virus. After it's bind to the AC2, the S protein changes its 3D structure completely to allow the membrane fusion between the membrane of the virus and the human membrane, allowing the virus to entry to the human cells. And when it happens, some patients can cause a severe acute respiratory syndrome. So it's the severe causes that is related with a formation of a blood clots that causes thrombosis in the lungs. That is very bad. And also a cytokines storm. That is a very bad inflammatory response in the severe cases of the COVID-19. So here's just showing the prefusion state of the spike protein, the viral attachment to the AC2 in the human cells, and after they bind, a lot of things happen to release one of the subunits of the spike to cause the fusion of the human membrane and the virus membrane, allow for a formation of fusion pore and the entrance of the virus inside the human cells. So the spike protein is one of the most important protein related with the invasion of the virus in the human cells. Here's just a representation of the COVID-2 has around of 24 proteins, shown here some structural proteins and non-structural proteins, and we are looking for the spike protein shown here, okay? So what do we know about the S protein? So the S protein forms homotrimers as shown in this cryo-EM structure. Okay, there are 3 copies of the same protein. And this protein is a very big protein with 1,273 residues shared in different domains as shown here, okay? One of them that is very important is the RBD domain colored in red. That is the receptor binding domain shown here in the up conformation, the other 2 in down conformation. That when it's in the up conformation, binds the human AC2 only in the up conformation. And here is the complex between the human AC2 and the RBD. Okay, this is a structure, crystallography structure, and this structure was also solved by cryo-EM, not for our group, for another group. But the spike protein has 2 subunits. The S1 related with the attachment, that is here, represented here, and the S2 subunit here down, close to the membrane. The virus membrane is located here, related with the fusion between the virus and the human, okay? So after the binding of the spike protein to human AC2, what's happened is like these 2 protease sites shown here, S1, S2, and the S2 prime, located here in the structure, is they are hidden by the protease. But when the spike binds human AC2, they change its 3D structure, exposing these 2 sites that are cleaved by a human transmembrane protease serine, TMPRSS2, that releasing the SU1 subunit that is attached to the human cell, allowing the fusion, as shown here, allowing to the fusion of the 2 membranes. So it's what, as far as we know about the spike protein. Okay. So when you look at close to the interface of the human AC2 and the spike, looking the residues related with the interface, it was very interesting because it is highly polar, related with polar residues, hydrophilic residues, and a few hydrophobic residues. So it is interesting because it is pop up in our mind that maybe this kind of interaction may be avoided by increasing inhibits concentration, because this is very common to be used in laboratory to avoid hydrophilic and salt bridge interaction between proteins. So we thought maybe if you add NaCl, salt, avoid the interaction or disrupt the interaction between the spike and human AC to avoid the entrance of the virus in the human cell, the replication, and the virus release. Based on that, we perform different assays using different concentrations of NaCl and salt to analyze the SARS-CoV-2 replication in virus cells. But first of all, we know that high salt concentration causes hyperosmotic stress due to cell dehydration and death. So it's a kind of, it's a problem if you use a high concentration of salt in cell culture, okay? So in order to face this problem, we perform different cytotoxicity assays shown here. So first of all, we use alarm blue cell viability cell, so we can detect the viability of the cells as shown here. So 110 millimolar, so it's the normal median culture, has already this concentration of salt. This is the control. Along the time, this is 0 hours, 1 hour, 24 hours, and 72 hours. So as you can see, the viability of the cells in the control is almost 100%, and keep like that until 1.7% in a cell. So based on this assay, the cell is viable in this range of salt concentration. But as we know that this is a problem, we did in another way, okay? By quantifying the LDH, it is an enzyme releasing the culture supernatant from cells with damaged membranes using this assay. So even this kind of assay to measure the viability of the cells are okay in all the conditions that we test. Actually, test more than that. So we saw cytotoxicity, so we are not shown here. So, but what I want to show is that in this range of salt, the cells are okay, they are alive. We also measured the percentage of dead cells stained with this, okay, to measure the DNA of the cells. And as you can see that in all range of salt conditions, from different time course with the salt until 27 hours, 72 hours, the cells are alive, and we only detect less than 80% of cell death in the NaCl range tested. In this case, after 72 hours, it detects less than 8% of cell death. So this range is safe to perform our assays. So we performed our assays in the salt range of 110 millimolar. That is the median culture that we grow virus cells. That is a kidney from monkey cells up to 285 millimolar cell in a cell. So what we saw is like that in two conditions that we test here, the post-infection and adsorption plus infection, that I'm gonna explain what's that, is when you treat the cells with salt for 2 hours and after you add the virus and keep it for 72 hours, or you add since the beginning the salt with virus and keep it for 72 hours, the replication of the virus is decreased. And in a way that is dose dependent in a cell. And in 1.4% of in a cell, we observed 100% inhibition of the SARS-CoV-2 using virus cells. That is very nice. But in another two different assays, that is virus pre-incubation, means that you put salt, different concentration of salts, only in the virus, and after you take it out, the salt, and you add to the virus cells, you not causes inhibition of the virus. Neither if you add the virus and the cells and keep the salt only for 2 hours, and after that you take it out the salt and put a normal medium, the virus still can replicate it. So the salt must be in the medium to avoid the replication of the SARS-CoV-2. So 1.4% is enough to inhibit in 100% SARS-CoV-2 replication in various cells. And based on our results, it's probably happened by an intracellular mechanism not related with the addition of the spike to the human ACE2. So based on these results, we can conclude that these data suggest that SARS-CoV-2 inhibition, the presence of salt, reflects an intracellular mechanism, and it is not ascribed to the dissociation of spike SARS-CoV-2 and human ACE2 complexes as we thought at the beginning. SARS-CoV-2 replication inhibition is NSA salt dose dependent. So it is very interesting, this observation. But another question is, so as we are wrong, our hypothesis was wrong, we performed some assays to understand how the salt inhibits SARS-CoV-2 replication inside of cells. So we conducted experiments to measure if hypertonic cells would cause a cell membrane depolarization. So what to observe is this: you will measure the depolarization of cytoplasmatic membrane. So as you can see here in the control, you have a little bit of depolarization after 72 hours, but increases when you increase the salt concentration. And this response is very fast. Each line is the depolarization of the cytoplasmatic membrane in different salt concentration. As you increase the salt concentration, the depolarization also increases. So the cell membranes showed a rapid depolarization stimulation with increasing salt concentration. After 15, almost 20 seconds, you are able to observe depolarization of the cytoplasmic membrane. This is dose dependent. The results of the 24 hours, you can see here in green. After incubation, all the time the salt is present. The depolarization of the membrane continues to increase with time. And after 72 hours, membrane potential tended to return to control, indicating that the various cells were partially able to recover the resting membrane potential. So what we can conclude is that the inhibition of the virus replication in the cell is related with the depolarization of cytoplasmic membrane. That is when you have the entrance of the virus. So probably it's avoiding the entry of the virus and the replication. How? So he proposed a mechanism on how this could happening. So we have the virus attached to the spike, to the ACE2, you have the entrance of the virus, and the normal cycle of the virus, you have the replication of the genome of the virus, and also the production in the endoplasmatic reticulum of different proteins of the spike, until you have the release of the virus by the exocytosis mechanism. So what do we think that is happening? So the NaCl concentration outside that we added causes membrane depolarization. Here, change the polarization of outside and inside of the membrane, or outlet and inlet of the cytoplasmic membrane. Causes an overflow of NaCl inside of the cells. That in the end causes a high ADP-ATP ratio, meaning that you have a very high amount of ADP, and ATP is essential for everything to the cell. It's essential for the RNA replication, is essential for protein production. So if you don't have ATP, nothing's happened. So how we can explain that? So the hyperosmotic extracellular NaCl concentration activates this pump, this NACS channel as a sodium sensitive to increase the intracellular NaCl. Also, recruit epithelial sodium NaCl, ENaC, shown here. Okay, so this also may happening. And interestingly, it had been reported that SARS-CoV, not SARS-CoV-2, SARS-CoV inhibits this channel. We don't know if it is also happened in SARS-CoV-2, okay? So it would cause an impairment in the mitochondria function, which it's meaning that if you don't have, you have a depolarization in the mitochondria membrane, you don't have production of ATP. That can explain the low amount of ATP in the cell. And another effect could be the in this pump, sodium, potassium, high activity to restore the potential of the cell. So they put out the inner cell that is inside, and this use ATP to happen. So this, in summary, what happened is that the cell is in a low energetic state that would be detrimental for the virus application. That is the mechanism that we believe that's happening, and you try to prove. What I can say until now is that we prove that the cell it is in a low energetic model. So we are trying to figure out if it's these channels are related or not. So based on this analysis, we have another part of the talk, is that the hypertonic saline solution, more than 1.4%, it's enough to inhibit SARS-CoV replication in various cells, also in lung cells that I'm not showing, probably by an effect related with the intracellular process affecting the virus life cycle. Hypertonic saline solution decrease respiratory syncytial virus infection. It was reported in the past, not from our group. And pro-inflammatory response, such as interleukin 6 and 8 release in culture of human respiratory epithelial cells, suggesting that this could happen with other respiratory viruses, the inhibition of the replication. And salt could be used as a treatment to different respiratory disease, because maybe this mechanism is a general mechanism. The complete molecular mechanism underlying the observed phenomenon may be more complex, involving several factors that together results in the inhibition of the virus replication. Therefore, further assays must be done to elucidate the real mechanism of virus inhibition by high concentration of salt. So the second question that we had, it is how SARS-CoV-2 spike protein keeps its interaction with human AC2 in low and high concentration of NaCl, since that the interaction between spike and the human AC2 is not disrupted in the salt concentration. So we perform different assays to measure that by computational simulation, also by experimental data using surface plasma resonance. So first of all, we analyze the sequence, only the RBD, between SARS-CoV and SARS-CoV-2, and we map the difference, the residue difference, shown in red, and what is in yellow highlighted, it is in the interface between the virus and the AC2, okay? So as you can see, the main differences are located in the interface between the spike and the human AC2 that's located here, and is shown here. And the characteristic of them change. So in the case of SARS-CoV-2 related to SARS-CoV, you have again of hydrophobicity, it's shown in blue, and again the hydrophobicity that is shown in red. So in this part that we call EU region, you have again alternate alternated gain, hydrophobicity gain, hydrophobicity gain, hydrophobicity gain, hydrophobicity gain in the E1 region. That's not happening in the E2 region. So it is shown that maybe you have two regions of the interface with different properties to bind the human AC2. So we named the SARS-CoV-2 interface in two regions, E1 and E2, as shown in here. That E1 has mainly hydrophobic contacts, mainly polar in both cases, but you have here in the E1 region hydrophobic cage, basically made by phenylalanine and this leucine, and each E2 is mainly polar interactions. So we performed a molecular dynamic analysis that showed two flexible regions in the human AC2 and the spike. When we observe this in the structure, here is this in the E1 region is flexible, shown in red, and in the AC2, it is in this loop far from the interaction that has two states. State 1, shown here in 100 millimolar salt, and state 2 in 100 millimolar salt, that goes from this open conformation to this closed conformation. And when you increase the salt concentration up to 1 molar, that's a lot, 1 molar salt, this try in human AC2 to have only one conformation, and in the spike, they also has a conformational change. And in both these regions of the protein, while you increase the salt concentration, they got more rigid, not so flexible, okay? So another observation is like while you increase the salt concentration, you have an opening state here, measured by the degree of opening the interface from 25 to 26 to 28 degrees here, suggesting that it is opening, but maybe not disrupting. Okay, so we also analyzed the covariance analysis, show all the contacts here in the interface. This is the human AC2 and the spike. So it's interesting that in the 3 different salt concentrations that we performed the molecular dynamics, you observe that 150 millimolar, you have a lot of covariance shown here in the interface. That is almost disappeared in 500 millimolar, that recover in 1000 millimolar. And they look like a little bit different, as you can see here in this displacement correlation matrix between the RBD and the human C2 interface. Here's the E1 region. You don't have a lot of interaction with human AC2 in 100 millimolar salt that appear when you go to 1000 millimolar. You have a lot of interactions, showing that while you increase the salt concentration, the interface is almost the same, but the contacts in the interface is a little bit different. So based on that, we suggest that the RBD E1, rather than the E2 region, becomes more firmly attached to human AC2 at higher ionic strength. Low ionic strength RBD E2 region becomes more firmly attached to human AC2. Then we investigated the relative contribution of these regions to complex stability in silico by applying harmonic force with constant velocity along the dissociation pathway. So we have 3 movies showing that. This is 150 millimolar salt. So we are looking for E1 region here and the E2 region. And in this case, I think I needed to get out to perform here. Let's see. So you can see here in the move that E2, GE1 keep it tight and E2 open first, 150 millimolar, while when you do in 500 millimolar salt, both regions open almost in the same time. But when you go for 1 millimolar, 1000 millimolar in a cell, the E1 keep like tight. E2 open and take more time for E1 to disrupt, suggesting that E1 it's like a hot spot in the interface between the human AC2 and the spike protein. So this is a very important part of the protein, the interface, to keep they both together. Based on molecular dynamics and cross-correlation, the spike RBD human acidosis is stable and a wide range of ionic strength. But is it also happening experimental assay? So it's what I have shown is by computation assay, but this is also happening by experimentally. So we performed surface plasma resonance in 150 millimolar to 1000 millimolar, and what we observe it's the same. The complex did not disrupt in this range of concentration salt. That's amazing. It's not at all, because it's a very low ionic strength to a very high ionic strength, and the KD, the dissociation constant, use only the RBD human AC2 do not change very well, neither if you use the trimer, the spike trimer, neither if you use the monomer of the spike or only the SU1 subunit. The KD, the dissociation constant in different range of salt concentrations, are almost the same. So different NaCl concentrations do not disrupt the spike human state complex. It's amazing. So what we can conclude in this the end of part of the talk is that at high ionic strengths, E2 mediated hydrophilic contacts become progressively weaker, while E1 mediated hydrophobic contacts increase to favor the complex maintenance. These features may reflect the necessity of the complex stability during release of the SU1 here, SU1 subunit, that accompanies the structural transition of the spike in this state, structure state protein, the viral attachment or prefusion state, to the fusion that's shown here, fusion intermediate state. So a lot of things happening here. You have different ionic strength happening all around the structure. So maybe the interaction between the RBD and the human AC2 must be stable in a very large range of ionic strength. So I would like to say thank you for Professor Edson Durigon from the Institute of Biomedical Science in the same department than mine, my postdoc Anacleto, Rafael Machado and Talita from the Henning department, to Hospital Israelista Albert Einstein from Brazil, Professor João Renato, Alessandra Leal for Gaspar Viana Clinic Hospital, Juliana Durigon from Medical School Clinic Hospital in Brazil, also a very big group from Institute of Chemistry in the University of São Paulo, Dr. Sandro Roberto Marana and his student José Davi Ribeira, and the company Acrobiosystem from China, who P&G performed the SPR assays. That I would like to say for the